Today I'm going to discuss the cell. The cell is a building block from which all parts of the human body are formed. I'm going to start by discussing the plasma membrane that goes around the cell. My cell is represented by construction paper. The plasma membrane here in black serves as a boundary between the cell and its environment. It allows materials such as water and nutrients to enter and waste products to leave. The nucleus is here in the middle in purple. The nucleus is our command center, and that's where we find our DNA. In the center here in black is our nucleolus, and this is where our ribosomes are produced. And our nucleolus is surrounded by a nuclear envelope, which is in red, and that acts as a membrane around the nucleus. Off the nucleus here, all in blue, we see our endoplasmic reticulum, and this is this has a series of folded membranes, you'll see all in here, surrounded in cytoplasm. It's the site of cellular chemical reaction, and it can either be rough with ribosomes, as you see here, these black spots represent our ribosomes, or smooth without ribosomes, as in right here, these kind of macaroni noodle looking pictures. Next, we will talk about the mitochondria, which are these orange oval shapes here. The mitochondria transform energy stored in food molecules into ATP. Has a highly folded inner membrane that produces energy storing molecules. Next, we're going to talk about the Golgi complex up here in green. The Golgi complex, or Golgi apparatus, it's a system of flattened tubular membranes that modifies protein and synthesizes them to their appropriate destination. And sends them to their appropriate destination. So this is like our FedEx control center right here. They package and ship proteins. They distribute the proteins made by the ribosomes. And then the vesicles we have in yellow here are like our little trucks that send them out. The lysosomes we have in brown here are our sanitation crew, because they break down particles or decaying organelle. And so you can remember lysosomes, Lysol, because they clean up the break down and clean up the decaying organelle. The centriole here in pink are a pair of small cylindrical structures composed of microtubules that duplicate during interphase and move to opposite ends of the cell during prophase. And then the cytosol is our, I'm going to add it back here, is our like gel fluid that's in the cell around all the organelles. And this is basically, this is your intracellular fluid, or ICP, and it's also, it's like a jello salad. It's very gel based. The peroxisomes, which are here in the blue, they resemble lysosomes, but they are not produced by Golgi complex. Rather, they are formed from endoplasmic reticulum. Peroxisomes detoxify the cells by producing hydrogen peroxide, which helps to break down unwanted particles and structures. Next, we're going to talk about the membrane transport, and basically there's several membrane transport mechanisms which allow systems to pass through the membrane. There are two ways to categorize. They have passive and active. And the active transport requires ATP, but passive does not require ATP. So several types of passive transport would be filtration, uses hydrostatic pressure to move particles. Pressure from water drives water and other particles actually through the membrane. Simple diffusion is movement of particles down movement of solutes from high concentration to areas of low concentration. So osmosis is a diffusion of water, the same principle of the simple diffusion from high to low. Carrier-mediated transport is moving through a membrane with use of transmembrane proteins. Two common examples are facilitated diffusion is one, passive mechanisms in which movement through a transmembrane protein without the use of energy, and then active movement through a transmembrane protein with the use of energy, or ATP. Vesicular protein or transport process used to transport large molecules across a membrane. Transportation of substances across a membrane in small packages called vesicles, which we talked about earlier here. And that requires ATP. Endocytosis is a process that moves substances into the cell by vesicles. Exocytosis moves substances out of the cell. We talked about the cell membrane earlier, and how it has two layers of phospholipids, also referred to as phospholipid bilayers. We talked about how the phospholipids are antiphilic. They have hydrophilic portion and a hydrophobic. Hydrophilic is a water-loving, and then you have hydrophobic is a water-fearing. We talked about substances that are allowed to pass freely through the membrane. Small substances, neutral substances, lipid-soluble substances made from fat, such as vitamins or steroids. And then substances not allowed would be large molecules, charged molecules, and ions.